Chapter 6 is over DNA, the molecule. So review questions. We've talked about this previously. What is a gene? So in science, the definition of a gene is not wholly agreed upon, and it's not a simple explanation. But what we know about genes is that genes are a sequence of DNA that basically is going to tell and provide directions for the cell to make and produce whatever the cell needs. Because each cell contains your DNA, they contain all the genes, but the function of the cell will depend upon the sequence of the gene that is directing that cell. So DNA is a short term or shorthand for the term deoxyribose nucleic acid. So you can see how we get the term DNA. A DNA molecule, as we know it, is a double helix structure, and it has two strands that make up a long string of nucleotides. Nucleotides we'll talk about in the next slide, but DNA is very specific in its arrangement in that double bond, or that double helix structure. It's basically like taking two strands that are wrapped around each other, or like twisting the red and white stripes of a candy cane. In the center of it, we call those the rungs. It's just like the rungs of a ladder, because if you were to untwist it, it would look kind of like a ladder. But the precise chemical structure of DNA is common to all life on Earth. DNA consists of nucleotides, and each DNA molecule has these subunits. So you have basically three parts coming together to form those nucleotides. You have a 5-point sugar, you have a negatively charged phosphate group, and you have a base that comes from one or two rings of nitrogen and carbon. So all four of the DNA nucleotides, which are adenine, guanine, thymine, and cytosine, we'll talk more about that coming up. They all have the same structure. The only thing that differs is going to be the DNA base. So the sugar and the phosphate group is the exact same. There's no difference there. What is different is in the nucleotides is the chemical of its base. So in DNA, there are specific pairing rules, meaning that certain DNA nucleotides can only pair with other certain nucleotides. So here we have at the very top adenine, which is only going to be able to pair complementary to thymine. That's a rule. So A is always going to go with T, and the first letters are just the shorthand versions. So sometimes that's how you'll see it sequenced in a DNA molecule. Underneath that, we have guanine and cytosine. They can only pair with each other. So again, the letter G is going to represent guanine, and the letter C is going to represent cytosine. So where they're held together, so you can see in between each of these nucleotides, that is called a hydrogen bond. And if you recall from the chemistry chapter, hydrogen bonding is the weakest form of chemical bonding, which makes it very easy to pull the DNA molecules apart. So just again, each strand of DNA is considered to be complementary to the other one based off of these pairings. So why is DNA such a great molecule for this hereditary information? Well, DNA, like we said, is considered to be complementary. So if you know half of the molecule, then you can build the other half, because that other half is just going to be complementary, which we can predict based off of the rules of the pairings. So in biology, as far as what happens in nature, is that the DNA molecule will unzip itself, and when we say it unzips itself, that means that the middle portion, the hydrogen bonding, is going to get pulled apart. And remember, hydrogen bonding is very weak, so that makes it very easy to unzip. Then what happens is, because it can unzip each of these strands can be used to make a copy. And that's very beneficial, because that means that the DNA strand is very quick to process in order to create a copy of itself. And so this is part of the reason why cell processes of mitosis can happen so quick, because DNA can copy itself so quickly. So during DNA replication, remember that a cell is going to duplicate its chromosomes. So this is when a cell is going from one cell to two cells and is making an extra copy of its chromosomes. What happens is that the original DNA molecule is going to be pulled apart and down the middle of the rungs of the ladder. That's where it's going to be pulled apart. And as it's pulled apart, the machinery in the cell is then going to create the complementary base pairs. So then what happens is, because it's pulled apart and the exact same base pairing should happen from the original, both of these molecules that you see at the bottom of the screen, hopefully that's popping up for you guys, both of these will be exact copies of the original. So one half of the original is used, and then the other is a new DNA molecule. So this is within the process of DNA replication. And this DNA replication is known as semi-conservative because it does not require a whole lot of energy for the cell to go through this process. So DNA is going to hold information on how to produce proteins. That is one of the ultimate goals of DNA. And so now we can talk about how DNA is used to make proteins, whereas the previous two slides talked about how DNA was going to be copied. So with this goal in mind, the cell uses a sequence in that gene that's found in the DNA. Once that gene is transcribed into RNA, it will then be translated into protein, and protein basically acts out the directions of what the DNA needs, whatever it needs that cell to do. So maybe that cell is going to be producing more hormones. Maybe the cell is going to be causing your nails to grow. There's the function of DNA within the cell, other than just creating more molecules of itself. So now we're going to talk about RNA. And RNA shares many similarities with DNA, but there are still some major differences. So the phrase RNA stands for ribonucleic acid. And remember that DNA stood for deoxyribonucleic acid. So RNA then is single-stranded, whereas DNA was double-stranded. So RNA is going to be a single strand, and you can kind of see a depiction of that on the slide. And here you would see the buildup of a few bases, like we looked at with DNA. However, one of the other major differences is that thymine in RNA will have been replaced with uracil. So a common way to recognize that you're looking at RNA is if you see uracil, whereas if you're looking at DNA, you'll see thymine. And another major difference is that because we're on RNA, the other major difference is that the sugar is not deoxyribose, it is just called ribose. So here's a picture to show you, for example, how this works. So we would start with a cell from this individual, and remember that in the cell is going to contain, or all cells of the body are going to contain the same sequence of DNA. That DNA is then going to be condensed into structures that we call chromosomes. If we uncoil the chromosomes, we will get the sequence of the DNA. So let's say that this person needs to grow more hair. Well, DNA then is going to direct a sequence of that in a gene. That gene is going to be transcribed into RNA, and then RNA is going to be translated into the protein. And then that protein is going to execute what the cell was going to do, which is grow your hair. And so that is what's going to help control a lot of your physical traits. So how this works in terms of genetic flow is that DNA to RNA to protein occurs in two steps, and the two steps are transcription and translation. So DNA, remember, is found in the nucleus inside of the cell. DNA then is going to be transcribed into RNA, still within the structure of the nucleus. Then what happens is that RNA molecule needs to be able to leave the nucleus, and it does so through the structure of the nuclear pore. And from there, it will then go out into the cytoplasm of the cell, where it will then find its way to a ribosome. Now remember, ribosomes can either be attached to the endoplasmic reticulum, or they can be free-floating. And at the ribosome is where that RNA molecule will then be translated into a protein. So step one is transcription. Transcription is taking DNA and turning it into RNA. So it's essentially taking DNA that cannot be read and turning it into RNA to be read. So think of DNA being in a language you don't understand at all, and then RNA is something that's close to English. So we could say it's maybe Latin, for example. And we could say that DNA is the language of Russian. And you might be able to speak Russian, but I'm using this as an example because I don't understand that language. So if DNA is in Russian and you don't understand it, it can be transcribed into Latin, and that would be your RNA molecule. Then what happens is that because of that base pairing in place of thymine, it's going to be transcribed into uracil. So all of the T's that you see on the left side will be replaced by uracil in RNA. What happens then is that the RNA molecule, which is specifically called mRNA, which is messenger RNA. This is just a version of RNA, and it's called messenger RNA because it's delivering the message from the nucleus to the ribosome for the protein to be made. So transcription's purpose is to make a molecule of RNA from DNA. So remember that DNA can separate pretty easily because it's held together by those rungs of the ladder, or those hydrogen bonds that are held together pretty weakly. And you are only going to use one side of the DNA to generate a molecule of RNA. Once DNA is done being pulled apart to make RNA, it will then coil back up. So RNA, for example, in a DNA sequence could be transcribed to do things like produce a hormone, cause hair to grow, cause the cell to get rid of any excess waste. Also remember that the DNA sequence that is being transcribed is a gene. So it's not the entire DNA sequence, it's just a small portion of that DNA sequence that we call a gene. And that's going to be making the RNA that will eventually produce the protein, which will carry out those instructions. So step two is translation. And translation comes from when you use the RNA molecule, and that RNA molecule finds its way to a ribosome. And the ribosome then is going to take it from a language sequence of mRNA to turn it into a protein. So remember that the M stands for messenger RNA, and it's going to find its way to a ribosome. And also as a reminder, these ribosomes can either be floating free in the cytoplasm, or it may be attached to the endoplasmic reticulum. So once it finds its way there, the mRNA strand gets read 3 bases at a time. And those 3 bases at a time are called a codon. And the sequence of a codon is going to correspond to a protein. So if we look at the sequence on the left, those first 3 ACU, just for simplicity's sake, we'll say that that responds into the protein in the shape of a triangle. UCC responds to the shape of a pentagon. GCU would respond to the shape of a square. So as the mRNA strand is going to be read, it will then build up into being this protein. And the protein will end up kind of looking like a pearl necklace, like you can see on the far right of the slide. Then it will be folded and then transported wherever it needs to go, whether that be inside the cell or outside the cell to do its work. So just to complicate things even a little more, in translation, there are actually 3 kinds of RNA. So previously in this lecture, we talked about mRNA, and remember that the M in mRNA stands for messenger. So that will be the name of the RNA that will go from the reading or transcribing the DNA or Russian in the language example into RNA for reading it into Latin. So mRNA is the name for that. The other two kinds of RNA are called ribosomal RNA or rRNA, and then tRNA, which stands for transfer RNA. So ribosomes are made from proteins, and they are also made from ribosomal RNA, which comes from the nucleolus. Transfer RNA are molecules that are carrying amino acids, and they carry those amino acids, which are your basic proteins, to the ribosome. So remember that your mRNA molecule is read 3 base pairs at a time into what we call a codon. So down in this picture down here, you can see we can divide them into 3, and you can see that the tRNA is going to read them. And so attached to the tRNA is a single amino acid, which, if you remember, an amino acid is a basic monomer of proteins. So one end of the tRNA molecule will contain the complementary codon that we will call the anticodon, and that matches up with the codon present on the mRNA. So how this might work is you would have a strand of mRNA, so say this line right here, and remember that this would be the Latin in our language example. And let's say that one of these sequences would be U, let's see if I can type on here, GC. And remember that mRNA will read 3 base pairs at a time. So then in order to read this, a tRNA molecule would come match it, and it would be read A, oh, that's not a great A, C, and G. And what would be attached to that tRNA molecule? Well, that ACG is going to indicate the correct amino acid. And the correct amino acid is exactly like the shape that we looked at 2 slides ago. So this could be the triangle, the pentagon, or the square. Let's just say that ACG is going to represent a circle. Well, that circle then is going to be an amino acid. In this case, it could be threonine, which is the example they have here. And that sequence is going to indicate a specific amino acid, which then, when linked together, will make up what's called a polypeptide, or also called a protein. So translation ultimately is going to result in a polypeptide. Poly means many, peptides always refer to a protein. So elongation or production of that protein is going to continue until the ribosome gets to what's called a stop codon. Stop codon is a 3 base sequence on mRNA that says that this is the end of the gene, and now you need to stop translating it into a protein. So remember that the gene is ultimately what's being translated here. So maybe this polypeptide protein is representing the production of a hormone, causing your nails to grow, causing your skin to turn over. That completed polypeptide is now available to be able to be used by the cell for its functioning. So remember that genes are what's going to be sequenced from DNA. So gene expression is when those proteins that came from the gene sequence on DNA is going to regulate what is happening inside the cell. Your cell can do what's called gene regulation, meaning it can turn genes on or turn genes off, and that is going to affect how your cell will function. So different types of cells are going to be functioning for different purposes. One example is that not all cells are going to need an enzyme called lactase. Lactase is used to break down the sugar called lactose inside your body. So if you don't have that, this function, so for example, digestive cells will need that function, then you're going to turn on lactase. But a nerve cell has no need to turn on the lactase gene. It has the DNA sequence to do so, it just will not because it does not have the function of processing lactose. So remember that all cells have the same DNA sequence and the same genes. It's just a matter of what genes get turned on or turned off. So in a nerve cell, the lactase genes would be turned off because it's just not needed in that type of cell. That process of gene regulation and gene expression is going to equate to the communication that occurs amongst cells. So there's three basic examples of how cell-to-cell communication is going to work when cells are going to communicate with one another. One of the key places is in cell-to-cell communication is vitally important for developing an embryo that is ultimately going to turn into a human being. So development is going to include lots of cell division that we call mitosis. That process of cell division is going to ensure that the organism gets bigger, and it's also going to ensure that the organism's functions of those cells be correct. So remember, you began as a single cell, and now you are a trillion-celled organism. And each of your trillion cells has a certain function and what it does for the body. So again, all of your cells contain the exact same DNA sequence, but each of your cells has a certain function. Another example of this is a mechanism for induction. So the cell-to-cell communication in the previous slide was one for development, and now we'll talk about induction, which is also very important to a developing embryo. So this induction is when a group of cells influences the development of adjacent groups of cells. And if its effects could be to switch genes off or switch genes on in those target cells. So for example, inductive signals can cause cells to change shape, to migrate, or even to destroy other cells. So in this case here, when your hands initially develop, they're all webbed together with the fingers. And then what happens is the cells between the fingers, they get the message that they need to die off. And that's ultimately what creates the space between your fingers. The cells that were once there to cause the webbing will have received the signal to turn off, and then they will die. You also have cell-to-cell communication that's called the homeotic genes. Homeotic genes are master control genes. So in a developing embryo, homeotic genes produce protein signals that turn groups of other genes on or off. Homeotic genes help to establish the overall structure of an organism. So for example, specific homeotic genes can direct which end of the embryo will be the head, while others can direct which body parts will be developing at which locations. So here you can see a mutation in a homeotic gene of a fruit fly. So the homeotic gene that has been tampered with is that in a normal fruit fly, you're going to have 2 wings, but in a mutant one, the fruit fly ends up with 4 wings. So those homeotic genes are very important to establish the body's axis. So far, we have basically talked about how your cell is going to be important for gene regulation and how every single cell of your body has the same sequence of DNA. And now I'm going to tell you that that's not always true, because mutations can change genetic information in a cell or virus. And you can assume that in you have many mutations, probably millions of them, but in you, they might not be an issue. Mutations, more often, can just be a simple base error and not cause anything to occur. So this may be something like a silent mutation, a nonsense mutation, a deletion. There are lots of varieties of mutations. These mutations can be spontaneous. So even when babies are born, they have not potentially been exposed to any mutation or mutagens, and they can still have mutations in their DNA. Or depending on your life activity, you can be exposed to what are called mutagens. Mutagens are physical or chemical factors in the environment that can change your DNA. So common examples may be smoking, a diet that's high in fat and cholesterol, exposure to x-rays, so like an x-ray machine or an MRI, even sunlight. So these things can cause mutations to occur. A mutation is any change in the nucleotide sequence of DNA So this could be replacing or adding a nucleotide base, which can have a huge range of effects on a DNA sequence. Mutations can happen naturally over the course of natural selection. So for example, when we don't have necessarily a good way to explain what occurred, so like dogs that have no hair and dogs that have hair, a common example we use for that is that there had to have been a mutation over the course of time that made that happen. Perhaps that's the reason that we don't need our appendix if there's a mutation, and then all of the succeeding generations from that first mutation no longer need their appendix, and that is just what evolved. So sometimes mutations can be helpful, many times mutations can be harmful. What might happen to the protein product if there's a change in the nucleotide sequence? Well, the protein would not be as it once was, and as a result, if the protein is not as it once was, it's not going to function as its original. It will be mutated. So for example, if your cell was creating or using a gene to produce, for example, testosterone, a hormone, possibly a mutation will cause it to then produce, instead of testosterone, it may produce a different type of androgen inside the body that's not testosterone. So that could be a mutation causing the protein to be different. So perhaps you know this, but a clear link to mutations can result in cancer. So the inability to control gene expression is cancer. Mutations oftentimes can cause a cell to not be able to control its ability to control mitosis. So mutations can lead to cancer. Not always, but it can. So in a normal cell cycle, like you can see it in the left, a cell goes through all of the phases of mitosis, so interphase, prophase, metaphase, anaphase, and telophase, and it has control as to how many cells will be produced and how often they'll be produced to make sure that there is not an overproduction. In cancer, what happens is that the cell will go through an uncontrolled cell cycle, meaning the amount of cells that's produced is not controlled. So this also does not control how often the cell goes through this process. And all of that comes from a mutation. Genes regulate the cell cycle, and so when the cell cycle is not regulated with that kind of light switch on-off scenario, it can lead to cancer. So a normal cell cycle system has basically a green light for go and red for stop. This helps to make sure that it's a normal cell cycle, and this ensures that the cell is going to be produced, that the cell is going to be making these new cells at very specific times, and it helps control this reproduction of cells. So an example of this is a proto-oncogene. So that prefix onco means cancer. So proto-oncogene is a protein that will tell the cell when it needs to duplicate. Proto-oncogenes are good. They're part of that normal cell cycle, ensuring that you have a limited amount of cells being produced, and it helps to stop cells from being overproduced. So what happens is that a mutation occurs in that proto-oncogene sequence. So let's say that the proto-oncogene sequence, if we were to say it's GCTA, that is a sequence for the gene of the proto-oncogene. What happens is that a mutation occurs. This could be by exposure to carcinogens such as smoking, sunlight, maybe it's just diet, but a mutation has occurred. And so now the proto-oncogene turns into what's called an oncogene, onco meaning cancer. So instead of being GCTA, the mutation will have caused it to be, say, GCAA. Now we have a cancerous gene. And when that cancerous gene occurs in the form of an oncogene, the cell cycle is now not controlled, which means that the cell can reproduce at a high rate, it can reproduce a lot, and there's nothing that is stopping it. And then this means that every single cell that comes out of this uncontrolled cell cycle has the same oncogene, leading to the cancerous accumulation of cells. So cancer is caused by out of control cell growth, and that's due to that breakdown of the cell control system. So cancer is defined as when a cell goes from having the proto-oncogenes, which are the normal genes to control cell cycle, to oncogenes. A mutation called the growth factor can produce that hyperactive cell to cause it to go through an uncontrolled cell cycle. A growth factor should only stimulate growth when it's appropriate, but a mutation in the growth factor can cause it to become hyperactive, meaning that it's not going to regulate its cell division. Another gene is called the tumor suppressor gene. So just as the name implies, it will suppress the development of an overaccumulation of cells. But in this case, when there is a mutation in that tumor suppressor gene, then the cell no longer is going to be able to be suppressed with the amount needed, and it will lead to an uncontrolled growth. So mutations can result in proteins that either don't stop a cell cycle, or they might actually stimulate growth. So cancer is that result of unregulated cell growth, and cancer begins with a single cell that has multiple proto-oncogenes that mutated into oncogenes. So it's usually more than one. The mutations disrupt the normal control of the subdivision and convert a normal cell into a cell that multiplies continuously and in an uncontrolled manner. If it's not destroyed by the immune system, because your immune system initially actually does a pretty good job of destroying any cells that are mutated, then what happens is that those mutated cells can then accumulate, and that is what forms a tumor. Cancers are also named for where they originate. So for example, lung cancer will start in the lungs, and if it metastasizes, then it can move to different locations. So also remember that proto-oncogenes are the good genes. These are going to help regulate your cells and make sure that you don't have an overproduction. When there's a mutation in that proto-oncogene, it turns into an oncogene, which is a cancer-causing gene. So when those cells form that tumor, the tumor is an abnormal growth of body cells. The spread of cancer in the cells of the body is called metastasis, and the metastasis can grow to different locations. Now, these growth at new locations is not necessarily bad, but if it's a malignant tumor, it means that it is cancerous and it is spreading to different locations. A benign tumor means that there's no spreading that goes on. Benign tumors typically are not fatal because they do not spread. Malignant tumors can be fatal because they can be spread. Most people with cancer basically have 3 treatment options, and commonly it's the combination of the treatment options, not usually just one. So surgery is an option because surgery can remove a tumor, and that might be sufficient enough to cure the patient if the tumor is benign. Other times you have to pair surgery with radiation or chemotherapy. Radiation is often localized and it's used for specific areas on the body. And you expose the body cells to that high level energy radiation. Energy radiation disrupts cell division, so it might slow down or kill cancer cells, but a side effect is that radiation can also damage normal cells. Now, chemotherapy, which you have probably heard about, involves giving a person drugs of chemicals, so it's literally means chemical therapy. A person is typically given intravenous drugs into the bloodstream, and the bloodstream then will deliver that chemotherapy to the rest of the body. And the chemotherapy then is working to control and kill off the cancerous cells. Common side effects, though, of that chemotherapy is that it might target more than one of the cancerous cells, and that can oftentimes destroy normal cells of the body. When that happens, that leads to side effects such as fatigue, hair loss, all kinds of nausea, because unfortunately, chemotherapy cannot just necessarily find and only find the cancerous cell. It oftentimes will find other cells and it will kill them. So what might happen to your normally dividing cells? Well, if your normal dividing cells go through this process and can potentially continue to divide, you still might have cancerous cells in your body even if you've done these processes. So as you know, there are ways to reduce your risk of cancer. So making sure that you're wearing an SPF, making sure that you're taking care of yourself with good diet and good exercise, getting regularly screened if cancer runs in your family or if you have suspicion. Not smoking. The unfortunate part is that really cancer is a matter of if those proto-oncogenes get turned off. So we all have proto-oncogenes in our DNA. Remember, proto-oncogenes are the good genes. They help regulate the cell cycle to make sure that the cell cycle is controlled. It is the transition from proto-oncogenes to oncogenes that creates cancer. And so in us, it's a matter of if it gets turned on, going from proto-oncogenes to oncogenes, or if it stays off, just like a light switch.